right, this video is on mitochondria and chloroplast, and we're going to talk a lot more about these and the specific reactions of respiration photosynthesis in Unit 3. So I'm just going to really touch on their structure and function right now. So mitochondria's function is to do cellular respiration, and their structure is beautiful for this. So they do have a double membrane. So they have this outer membrane, and then they have an internal membrane that is convoluted or folded. Those folds are called cristae, and they're going to increase surface area because the electron transport chain occurs along that membrane. So the more of that membrane you have, the more electron transport chain you can do, and the more ATP you can make, which is the whole point of the mitochondria, is to convert food energy into ATP. And so that's a direct link right there between structure and function. The fact that they have two membranes creates a space between them. That space is called the intermembrane space. It is going to allow for some concentration gradients to be formed. So when we are doing the electron transport chain, one of the things that we're going to do is we're going to pump hydrogens from the matrix out into this intermembrane space until they get a really high concentration, and then they'll fall back through the membrane into the matrix. And when they do that, they will form ATP. That will give the energy for ATP to be formed. So that's one way that the structure is related. The matrix is a fluid-filled inner compartment. It's kind of like the cytoplasm, so it's not liquidy, but it's not solid either. It's a kind of a jello-ish, jelly kind of structure. And it is where your citric acid cycle, or what we used to call the Krebs cycle, occurs. And cycles generally occur in these kind of liquidy environments because it is what works best for the enzymes and all that are involved. It allows everything to run more smoothly. And so matrix is set up for that cycle. The intermembrane with the cristae is set up for the electron transport chain. And so definitely structure and function are linked for these two because it is perfectly suited for its job. And this is just kind of showing you. So this is the intermembrane space in here. You've got your Krebs cycle happening in the matrix and your electron transport chain system occurring along this membrane. All right, chloroplast. Its job is photosynthesis. It is also double membraned, just like the mitochondria, and we'll talk next week about why that is. There are thylakoid discs in that. Those are stacked up in things called grana. And there is also around this, so outside of the thylakoids, there is stroma. And so stroma is that fluidy, jello-y, jelly-ish type of structure. And it is going to be where a cycle takes place, just like in the mitochondria. All right, so when we zoom in on a thylakoid, I don't know if I have a zoomed in picture, but you can kind of see here, the thylakoid is kind of like an empty sac of itself, and it has a membrane. And it's not empty, there's a fluid in there as well, or a semi-fluid in there. Anyway, this membrane is where the electron transport chain takes place. In this case, now we're going to pump hydrogens from outside the thylakoid to inside the thylakoid across that membrane. It's going to allow us to build up hydrogens in the inside of the thylakoid, and then they will fall back through the membrane, and that will form ATP. That ATP is then used to form glucose in the Calvin cycle, or the dark reaction, which occurs out here in the stroma. So the function of the thylakoid is to convert sun's energy because it's receiving sunlight. That's why it's green, because it has chlorophyll. It's receiving that sunlight and converting it to ATP, but it needs that membrane to do that because it needs to be able to form that concentration gradient. So structure tied to function. Then two things that are formed from that are NADP plus and or NADPH and ATP. Those are going to be right because they're both formed on the outside of the thylakoid, so they are going to be right in the stroma where they need to be, so just the space around the thylakoids where they can then do the Calvin cycle to form that glucose. And so that is how the structure of the chloroplast is related directly to its function of photosynthesis. I hope that helps, and if you have any questions, let me know.